So my name is Andrew Hung. I'm from RMIT University in the chemistry department. And my area of research expertise is on computational modeling of proteins and peptides using high-performance computing, especially focusing on the design of antibacterial treatments, including peptides and molecules, as well as using computational methods to solve problems in neuroscience and other physiological disorders. So this particular project involves the discovery of effective antibacterial peptides derived from the octopus genome. And this is a work in collaboration with my longtime colleague and collaborator and friend, Wenyi Li.
My name is Dr. Wenyi Li, a senior lecturer at the Trobe University at Department of Biochemistry and Chemistry, and the Trobe Institute for Molecular Science. For this project, we started because the bacterial infections, so antibiotic resistance is a big issue. So antibiotics, a lot of us are using or experienced with that. So since the discovery of penicillin in 1930s, and then they isolated penicillin and then used quite a lot for Second World War, saved millions of lives. And afterwards, antibiotics has been becoming a golden age, and then have a lot of different antibiotics has been discovered by a lot of scientists all over the world. So in 2014, there's an O'Neill report, and so they have already predicted there will be around 10 million deaths in 2050, which is directly associated with antibiotic resistance. What we are working on, especially for this project, so we are working on peptides.
Peptides and proteins are really just sequences of amino acids, so we can treat that as a language. We can treat that as a sentence. The amino acids might be words, or maybe clusters of amino acids could be form some sort of a sentence fragment. So they have a grammar of their own.
We are working on it's called antimicrobial peptide, but also there's another name which is called host defense peptide. Most of them are existing in nature, in most of the organisms, so they is contributing their defense system of the organism. A lot of people working in antimicrobial peptide development, trying to use this as an alternative for conventional antibiotics we are using. For this project, we are interested in what we are more likely to find out, because there's a lot of protein in the resource. So there's other ways that you can find the host defense peptide or antimicrobial peptide you modified and then get new ones. But we're also interested in trying to find from the protein database or from this project, which is Octopus, they have different protein fragment, which people didn't report. They have all protein, they have the antibacterial activity. What we are interested is we trying to see if we can find the protein fragment, which is the small peptide, from the protein database, trying to use the artificial intelligence, the prediction tools, or trying to see if they can predict that the certain of the peptide has the antibacterial activity, and then we can work in the wet lab.
So from much of the work that I carry out is in close collaboration with experimentalists such as Wenyi, and for my other work involving other classes of peptides, such as those from cone snails and snakes, I work with other collaborators who are interested in other research problems as well. So as well as antimicrobial aspects, there are also many of these targets, many of these protein targets are also involved in other physiological processes and diseases. So a lot of the choices of the peptides, what types of peptides we use, are driven by the experimentalists, and part of that is really the readiness of acquiring these peptides from nature. So, for example, just to maybe talk about the cone snail just very briefly, we had collaborators who actually go deep sea diving and hunt for these cone snails and extract the venom. So a lot of the sources actually are driven by the biological availability of the peptides, but also from the computational aspects, structural information as well. So a large part of the computational work is driven by experimental data. So if there is existing experimental data for what these peptides look like, what are their atomic structures, what are the molecular structures, then we would tend to select those peptides and proteins to do our computational studies as the fundamental foundational work to develop other new proteins and peptides. Well, one of the most exciting aspects of the computational aspects is that we can actually identify which parts of the peptide may be responsible for its activity, antibacterial activity, and that information, which we can extract at a very fine, very high resolution level, even down to the atomic level, gives us a lot of opportunities for further collaboration, between myself and Wenyi and other labs to actually improve these peptides. And this is all drawn from the computational picture or the computational structures that we can obtain from these simulations. One of the things that we actually extract from our long-standing collaboration in this and other projects is really to gradually build up a sort of a knowledge base of how these peptides interact with various biological molecules involved in bacteria, so including membranes and in this case the DNA, and gradually to build up a general model, a quantitative model that can help us make further more accurate predictions that can help save time in terms of helping us decide which particular changes may be most valuable in terms of enhancing the function of these peptides, as well as making them more safe. So we all, not only do we want these peptides to be able to target bacteria, we also they don't want to be harmful to humans. So this is the general quantitative model that's we're developing as we gather more and more experimental data from Wenyi's lab, as well as the computational data at the molecular level from our work. I think there are two aspects of AI that was used in this particular project. Although AI is expected to be a larger part of our overall project in future, in a more focused way in this particular project, the first way that AI was used is really the use of external servers, database servers that's publicly available, to scan through the octopus genome to identify potential antimicrobial peptides or short peptide fragments that may exhibit biological activity. So many of these servers use deep learning methods, neural networks, various other machine learning approaches. So that's one aspect in which AI has been incorporated in this work, the initial discovery of the peptide sequences. The second aspect is structural. So in the molecular modeling and simulation, the structure of the peptide, the octopus peptide, was constructed with a deep learning structural prediction tool called AlphaFold. Yeah, so just to sum up, the first is to look at the sequences, applying AI methods to scan through the sequences to identify the peptides. And the second part is to take the sequence that we've identified to build a 3D structure.
For us, we work in the lab, so we really see AI is a very powerful tool for drug design, or even give more the information about the discovery or prediction. I think that's another direction in the AI tools, and how they predict. They can change the peptide and they can change the protein sequence, but how they can predict when they interact with their target, how predict their structure change, because that's probably more information to how they interact with their targets or bacteria. That's probably even more powerful for the drug discovery.
I see that there's a really big role that AI is going to play in all aspects of research, including peptide design and understanding peptide interactions. Where that's going to go, I'm not too sure, but wherever it is, it will be something that's I think will be quite intuitive to use. That's will open up this field to many more people.
are trying to building a big library, trying to see if we can to combine with the virtual library and also our currently tested the lab data to have a more precise prediction tool.